Everybody get signed into the Kahoot. We're gonna give just another minute for the people who just joined online to get signed in. We'll talk about what's going on and what we're gonna be doing once we do this poll. We're gonna go ahead and get started. It's just a poll, so this is just not anything crazy yet, okay? It's gonna ask some questions. Pick the one that applies, one, or if there's multiple, select everything that applies to you. There's only 6 choices. I made the time longer. It's okay. So most people have completed the identity verification and have had access to Canvas restored. 73% of us have. We have people that have submitted their identification, but they're still waiting for access to be restored. This is something that, again, our IT was not open this weekend, but I believe they're pulling weekend hours to try to help with activating this. So if you're still waiting on that, I have had access to the Canvas app, but have not been getting emails or announcements. Okay, for those people that have access to the app and have not been getting emails or announcements, if you selected purple on this, how many of you have not started your identity identification process? So at least if you don't have that emails and you haven't been getting announcements, you have started the identification process, yes? Okay, it says 9%, I don't know, this is 2 people, have not started identity verification yet. But you knew we weren't going to have a test today. Yes? Okay. So for the 2 people that did not start identity verification, I know there's 1 person that is waiting on their ID. Yes? That's yes. So we know that there's that. For the other person, do it. This is not optional. Get it done. Get it started. Okay. So if you haven't done it, there's announcements. There's like several announcements on Canvas. Just read through the announcements. If you're like, I haven't been getting the announcements or the emails, read the announcement. I got quite a lot of emails being like, are we having the exam? Blah, blah, blah. And I'm like, read your announcements first. If you didn't find your information in the announcements, then, and if you're like, I wasn't getting, emails about it. Yes, I understand. But if you had access to the app, you could have gone into the announcements and read the announcements. Yes. Okay. So that was kind of a little bit one of those things where I was like, I was getting a lot of emails about stuff like that, where I was like, I want to help you out. I want to make sure we're on the same page, but at the same time, like, read your announcement. If you don't know what's going on and something weird is happening, check the announcements first. If you had access to the app, it should have at least been on there, even if you weren't getting the prompts or the emails for it. Once you get your access to Canvas restored, then you will be able to reset your password for your email and the things like that. So you can get the emails and stuff like that. But again, you'll have to reset your password and all that stuff. Are we all clear on, if anybody hasn't started the process for identity verification, are you clear on what you need to do for it? Yes? Okay. Yes, Daniel. Yes, so you have to do that. That's not optional. So that there is information on there on the announcement. There should be several announcements on what link you can use to start that process. Because again, if we're sitting here saying like we have access to Canvas, you mean the app, right? If you log into the computer, it won't let you log in. So again, where we take our exams on the computers, right? Not in our phones, on the apps. So that's where the issue is and why we can't have the exam today. When we've only got 73% of our students, that wouldn't be fair to the other 27% that don't have proper access to Canvas once we start the exam. I mean, so we've had people that haven't had access like all weekend to be able to study for their study materials. So we're not going to have the Unit 2 exam today, obviously. Most of you guys are aware of that and knew that that was going to be the case when you came in here. Because we have so many of us that have started the process and will have access again. How many of you guys have been without access the entire weekend? Well, we've been able to at least access on the apps. Has that been okay? It's been accessible. Then we're having the lab exam on Wednesday. Okay. That's not going to be postponed. All right. And if for some reason you're one of those people that's like, I still like can't sign in on the computer, but I can access the app, then I'll give you a paper test if it's not restored by then. So Wednesday will be the lab exam. We're still going to have the lab midterm on Wednesday. We clear? Okay. So that'll still take place. We won't have to postpone that. It's just not worth it for me. I say that now, but keep a lookout on the announcements. Again, you might have to manually check them if you don't have access yet. If I change my mind about the lab midterm, because it depends on the other classes too. Like maybe 73% of you guys have it done, but what if my other classes, like not even 50% of them have it done and they haven't had, I don't know. And so that's the problem with this, right? And so if you're sitting there thinking, wow, why didn't you guys plan this better? Why didn't somebody do this at a better time? Obviously, we're in midterms week this week. Why didn't they plan this? We didn't get a choice about this. This was an FBI mandated reset of our password system and verification of identity of students. So we didn't get notice on purpose because we didn't want fraudulent students trying to make a way to get around the system somehow. Because this is all about fraudulent students. There are, you know how OSU OKC, like once you are accepted or whatever you go through the admissions process, which is basically just submitting basic identification. You don't even have to show up in person to sign up for classes at this school, right? You don't. And there are students, quote unquote, that have signed up for classes and are applied for and receiving government benefits. And they're getting money for, tuition reimbursement and things like that for classes that they've never shown up for. This is so much of an issue that the FBI got involved. And we have to actually validate the identity of every single student that's enrolled at your university. Doesn't impact classes like this as much because we have prereqs that you have to show that you've got your credits in order to enroll in my class. But imagine like Comp 1 and things like that anybody can sign up basically to take that. And I'm telling you, it's not a small problem. It's something that we've been talking about for years, but has just been now so much of an issue continuing that it has, we've had to shut down our system. I had a lot of students reaching out and being like, why wasn't OSU Stillwater affected? Because they're not an open enrollment university. They do have to you have to go through an actual advisor. You aren't allowed to just enroll in classes, that sort of stuff. So Stillwater doesn't have to go through it because they have to go through the identification process that way. But here, like people can be completely online and never have met a person face-to-face and just enroll in classes and get federal funding, reimbursing them for those classes that they never attend. And so anyways, and if you don't think this is a real issue, I'm telling you, I've had students tell me that they have family that are in the prison system that have asked for help with this sort of thing. So yeah, so that's why this happened. And that's why it happened as quickly as it did and why we didn't really get any choice or notice about it. We got maybe 20 minutes notice before it happened and why we're having to do this way. So bear with us while we do that. We're going to postpone the Unit 2 exam. The Unit 2 exam will be the Monday we return from spring break. So that day we will be just doing that. Odds are we will probably not have lab that day either. So we'll have to probably postpone that lab or absorb it into other labs. Just because my online class, they don't have a separate lab time for the exam time. You know what I'm saying? So Monday, just so we're aware, today we're going to do some review stuff. Wednesday, we're going to do the lab midterm, and then you'll have spring break. Monday, when you return, we will have the Unit 2 exam. And right now, the dates in Canvas do align with that. And so there's that to be aware of. For Unit 3, because this is a problem, because on Monday, when we return from spring break, we're supposed to start Unit 3. Our first 2 chapters for Unit 3, chapters 11 and 12, you're going to have to be responsible for it, and I'm going to assign it as hybrid. I'm not going to be able to delay it. We do not have the days for that. So you're going to have to start Unit 3 before we start the Unit 2 exam. And it is what it is. You deal with it how you want to. If you just want to knock out the questions for the Unit 3 lecture, you can do that, but it is going to be due the Sunday before we come, that Sunday. So be aware of that. So those will be, again, that assignment has not been made yet. That's my responsibility. I was planning on doing that this weekend, but I've been dealing with this windfall. It's been a lot of work managing this. So we will get that updated. I will get those assignments made and whatever. So again, over spring break, what you'll be responsible for is starting that Unit 3 stuff and studying for the Unit 2 exam. And then on Monday, we'll come and just do the Unit 2 exam. We will not have an in-class session for the first 2 chapters of Unit 3. Those are the 2, I feel like 2 of the easier chapters too. So it's mostly like just memorization. It's not a lot of, I don't feel like too heavy on the concepts. So I think we'll be okay with that. But I don't have the days to like extend that out and have time on that. Do we have any questions about any of that? Okay. So again, if for some reason we're not going to have the lab midterm, keep looking out on the announcements for that. Because I think everybody has at least access to the app, right? Okay. So even if you're not getting the emails. And if you did get verified and your identity was verified and you changed your password and stuff, you will have to, if you are using Outlook for your email, then you'll have to reset your password in Outlook for it to start sending you emails again. Otherwise, if you're just doing the inbox thing on Canvas, it should be there when you get emails. All right. We're going to do, okay, let's ask you guys what you want to do. We're going to do the lab midterm on Wednesday. Would you rather review for Unit 2 or review for the lab midterm? Okay, so that's going to be the question. Hands up if you would like to review for Unit 2. And hands up if you'd like to do the lab midterm. Okay, let's do that then. I figured. Yes. All right, just double check that you're signed in on this. And we'll do this review, and whenever we get done with it, we'll be done. It might take the whole class period, it might not. We're not going to have like lab today. Okay. So whenever we get done with this, we'll be done. So at least there's that. All right. Okay. Let's get into the mindset of the lab stuff. You might have already played through this one, or at least walking through it, we can cover a little bit more information. Part of the lab exam might be stuff about safety, so keep that in mind, but it will be very basic stuff about the lab safety. That you have to wear a lab coat and closed-toed shoes, that you should put anything that is made of glass, that is, any slides or broken glass goes in the sharps container. Otherwise, things like disinfecting and stuff like that. So basic stuff. It's not going to be complicated questions about lab safety. It's going to be very basic. Because also on the lab midterm, which is on Wednesday, you must have your lab coat and closed-toed shoes. Otherwise, you'll get points off on your exam. Okay. So be sure on Wednesday that you do have your lab coat and closed-toed shoes for that day. Okay. All right, where should broken glass be disposed? Pretty straightforward. There will be some questions on the lab exam I'm not saying it's a lot of them, okay? But there might be a couple that are this easy. Okay, so it's just, again, basic application there. All right, so starting off with our stains. You should know the steps in order, but you don't have to know like the details of like how long it has to sit or anything like that. But again, if we're talking about the gram stain, you should know that we start with crystal violet, then we add iodine, then we would rinse with the ethyl alcohol, and then we would do our counter stain with safranin. So you should know what stain you're going to be using, in what order, and what purpose each step serves. So could you explain to me, what does ethanol do in the gram stain? For example, you would know that it's a decolorizer. It'll remove any crystal violet that did not get fixed in place, for example, okay? It's all multiple choice, but at the same time, you've seen my multiple choice choices. A lot of times, some of the answers, if you don't really know your stuff, you might get confused in it. And also, y'all have a tendency to overthink at a level that is very impressive. So So be sure that you know your stuff well enough that your overthinking doesn't get ahead of you. So know the stains and their purposes. For each of the stains that we did, do you know what a positive looks like and what a negative would look like? And could you identify the positives and the negatives if I gave you a mixed sample? And what happens if you skip a step, especially in major steps like decolorizers and things like that? What would happen if you skipped those steps and what would your samples look like? All right, so let's get into it. Select all that apply. What is the purpose of heat fixing? What does the heat fixing itself actually do? All right, good. I'm so glad that I don't have to explain why it's not stain it, right? So, okay, so it adheres it to the slide and kills it. Great job, guys. So it does kill the sample and adhere to the slide. We heat fix in a way that, yes, the organism dies, but we're not denaturing it fully. We're not changing it so much that we're like, incinerating it, right? So that's the whole point of the heat fixing. And heat fixing process itself obviously doesn't stain it, but does it prepare it for staining? Pairs it for staining, right? Okay, good. What is the counter stain that we use in the gram stain? Counter stain for the gram stain. This might be an example of an image of a gram stain. I feel like there's enough information on this image to tell you that the organism is a gram negative in that image. So the counter stain is going to be the one that again, counter stains are always there to color negative cells or to color decolorized cells, just to provide contrast. So what's our primary stain in the gram stain? Crystal violet's the primary stain. Then we have iodine as the mordant, and then we have our decolorizer is ethanol, and then we counter stain with safranin, which is our red. And in this image, you might be looking at it and thinking, those are purple, but look at the purple blobs. You see what I mean? And so there's enough information in the slide to tell you, okay, the purple's probably stain artifact. And overall, the cells are they're pink, but we stained them with the red stain to get that color. So this is gram negative organism here. So stuff like that. And if you were to ever look at this in a microscope and you'd be like, I can't tell if this is pink or purple or what, you can always ask me. And I would prefer questions like, I think this is red, as opposed to just, what color is this? Try to get some context, but I'll still help you if you're like, what color is this? But try to get some context, and I'm going to be more likely to give you a little bit of a nudge if you give me some context, okay? All right. Next, what color will a gram negative bacteria be after the gram stain? Kind of just said it, but gram negative. All right, perfect. So it's red. And we will see purple for gram positive and red for gram negative. And so those are our two positives and negatives for the gram stain. Keep context too on what that means. If we stain red, it means you have a thin wall and an outer membrane. If you stain purple, it means you have a thick wall, right? Okay. Next question. What is the first stain used in the capsule stain? Capsule stain. Alrighty, so so the capsule stain is the one that stains the background, and we only used one background, also known as a negative stain. So remember, negative stains stain the background, positive stains stain the cells. And so the only negative stain that we ever used was in the capsule stain, and it happens to be our first stain that we use in the capsule stain. It's our primary stain, and that is Congo red. Congo red is our first stain in the capsule stain, and it is a negative stain. It creates the background for us. Our secondary stain in the capsule stain is Medieval's, and that's really the only two steps that we have. We have the Congo red that we mixed with our sample, then we spread it out, then it air dried, then we added Medieval's on there, and then we let it sit for a minute, and then we dumped it into our dunking jars, the Copland jars. And so that's the steps of the capsule stain, and we looked at it in a microscope. All right, let's move on. We're going to talk about the other ones in a minute. Select all that apply. What type of stain is the Congo stain? Select all that apply. I just said this one too. Okay, so negative stain, background stain. So negative stain, stain the background, okay? So yes, I feel like that's fine. No need to get more complicated than that. Negative stains, stain the background. Congo red is a negative stain. All right, this is a pure culture that's going to be pictured. Which stain is pictured? And if you were to ask me what colors are on this, I would say a greenish teal color and red as well. This is a pure culture. So we also have to know what a pure culture is. All right, so good. So this is the endospore stain. If it's a pure culture, that means it is one species of bacteria. And so don't fall into the trap of being like, oh, this must be one species of bacteria. Therefore, this must be a pleomorphic bacteria, because that's not what we're seeing here. This is not a pleomorphic bacteria. Remember, pleomorphic means many shapes. So we're looking here, do we see different shapes? Yes, but the rods are the vegetative cells and the green are the endospores. And if you're looking and saying like some of the rods have green on them, yes, because those are sporangiums that are forming endospores. So we remember those terms, right? So this is the endospore stain. And so we have stained endospores throughout here. And the images that we saw in lab, again, I wasn't there that day, but the images that you were able to see and the images that I put up on the board had quite a lot more endospores in it, but it looked very similar to this, right? It had a lot of the bubble shapes around everywhere. So remember, you'll see an actual slide on a microscope in the lab exam. It won't be pictures. Okay. What is the term for those red structures that were in that same image? All right, so that's the vegetative cells. If we were talking about protozoa, trophozoites would be the word for the equivalent. But remember, vegetative cells, we use that term for bacteria. Trophozoites are only used for our protozoa, okay? But our active multiplying form of our bacteria is called our vegetative cells. All right, next question. Which of the following bacteria would be acid-fast positive? Verification, it's not polymer. All righty. So this is the type of a thing where you might have a few questions that are like this. All of like the stains and the procedures that we did for the bacteria, the ones that you should be able to identify based on like a stain result. The acid-fast stain tests for mycolic acid, which is wax in the wall. What primary stain did we use to perform the acid-fast stain? Does anybody remember? Carbylfusion. So it's lipid soluble and it will bind to that mycolic acid wax. Again, mycolic acid is found on the surface of Mycobacterium species, Mycobacterium. A lot of us picked Staphylococcus aureus on this. So this shows me how much we haven't studied for the lab midterm that is coming up on Wednesday. Probably because we were assuming that we wouldn't be having it, but there's so many of us that have access to Canvas, at least to the app, that there's no reason to postpone the lab midterm now. So keep that in mind that the lab midterm is coming on Wednesday. So Staphylococcus aureus, what would it be acid-fast positive? Should be acid-fast negative. Only Mycobacterium species have mycolic acid wax, and the acid-fast stain is looking for mycolic acid wax using carbylfusion. Okay, that's the idea here. So it's not going to be that one. It will be this one here, Mycobacterium. You don't need to know that. You don't need to memorize that it's going to be flea or anything like that, because it could be Mycobacterium tuberculosis, Mycobacterium whatever. But if it has Mycobacterium in the first part, right? Okay, one of these other guys, and just one of them, would stain positive for another staining procedure that we have done, which are differential stain for the endospore stain, and that would be Bacillus subtilis. So remember, Bacillus or Clostridium, will stain positive in the endospore stain. Any bacillus or Clostridium. And then Mycobacterium is the acid-fast stain. The other stains that we have done, the capsule stain, most bacteria have a capsule. And the gram stain, you're either positive or negative, but it doesn't help you to identify necessarily species that way, right? But the acid-fast stain, the endospore stain, you can cut it down to a genus, just by being positive for that. Okay. So have a little bit of reference there as well. All right, what is the primary stain that we used in the acid-fast stain? We just said it. Let's see if you can recall if you were paying attention. Acid-fast. I don't remember if I have another question about this one. But we'll talk about it anyway. All right, so this is carbofuscin, which again binds the mycolic acid wax, which is a lipid. Carbofuscin is lipid soluble. All right. What color would you be if you were acid-fast negative? What color would you stain in the acid-fast stain if you were negative? Anybody remember? Blue, yes. The counter stain is going to be methylene blue. So you stain with your carbofuscin, which is fuchsia, and then you would let it sit for 5 minutes. So you don't need another 5 minutes. Then you would rinse it with acid alcohol and then counter stain with methylene blue. And that's the process for the acid-fast stain. Good. A lot of times on the lab exam, I give a mixture. There's a picture in your lab manual of it. Okay. I often give a slide that is a mixture of positive and negative. Because again, in real life application, if you have somebody that has a cough and you think they have tuberculosis, in my lab at Integra Southwest, every single day, we would get acid-fast stains for sputum. And is sputum just going to be only positive or nothing in it? No. So we're going to be seeing a lot of cells no matter what. But if they're all blue, your patient doesn't have any mycobacterium. But if it has any of the fuchsia, the pink, then that patient has tuberculosis. And to be clear, mycobacterium species, there are some species that can be part of your biota, but not in your lungs, ever. And so if we see any acid-fast bacteria in sputum, then that is an indication of it being tuberculosis. There are two types of bacteria that can cause tuberculosis, primarily Mycobacterium tuberculosis, but another one is Mycobacterium avium, but they're both Mycobacterium species. So they would both be positive on this. All right. In which staining procedure did we use steam? Why there's no music on this one? Makes it feel real, I guess like the test will be, it's gonna be real stressful. All righty, so the procedure that we use steam on is the endospore stain. So this is the one where we had our slides up on top of our beakers while we were boiling the water. The day that I wasn't there, that you guys had Dr. Tim. So we used steam, and in this endospore stain procedure, just to review that one, right? Our primary stain for endospore stain was malachite green. Our mordant, and there was a mordant, was the steam. Then we rinse it with water, which was our decolorizer. Water actually acted as a decolorizer in that one. And then we counterstained with safranin. So those are like the 4 steps for the endospore stain and how we get our vegetative cells to be red and our endospores to be green, or to be green. All right, so that was the endospore stain because the steam works as a mordant in that procedure. Good. All right, which of the following structures allow for bacterial motility? What do bacteria use for motility? All right, so this is flagella and only flagella. So eukaryotes can use flagella and cilia and pseudopodia, but bacteria only use flagella. They use fimbriae for attachment to surfaces. They use the sex pili for the conjugation to show DNA between the cells. And bacteria do not have cilia. Good. All right, so the simple stain. Don't forget that the simple stain is a thing too, okay? So if we're doing a simple stain using just methylene blue, like we did in our lab whenever we did lab 2, right? We just used methylene blue. We made our heat fixed slide. We stained it with methylene blue. all cells, no matter if they're gram positive or gram negative, or if they're eukaryote or akaryote or anything. If you're doing a simple stain with methylene blue, all the cells will come out as blue. It's not differential. It doesn't tell you differences or anything like that, right? All right, gram stain does depend if you're positive or negative. Positive is purple, negative is red. And we already went through the steps for that one. The acid-fast stain tells you whether or not there is mycolic acid wax in our thick walls. Remember, all acid-fast bacteria are gram positive, so they all have thick walls. And so the acid-fast procedure, again, is carbofuscin, and then rinse with acid alcohol, and then counter stain with methylene blue. The capsule stain, this one, again, we're looking for the presence of no color, is really, we're looking for a halo of no color. It looks white in the microscope if you have a little bit of white around the cells. But keep in mind that in the capsule stain, it's like a purplish bluish background, typically. And then the cells themselves look like reddish pink. And then if you have white around them, then that's the capsule. Because again, the capsule is a densely associated thing for adhesion, for evading phagocytosis, and for creating biofilms. Those are the possibilities of functions of the capsule. And it won't stain with those stains that we use in that procedure. So carbofuscin, not carbofuscin. And the staining procedure for the capsule procedure is going to be Congo red as your background stain. And then Meneville's that changes the color of that as well as the cells. And then colorless would be the halo of colorlessness is the capsule itself. The wet mount, we didn't spend a lot of time on it. You're not going to get tested a lot on it either. But two things you should remember about the wet mount. I guess there's a couple of things we can go over. Number one, it shows you whether an organism is motile. If it is motile, then you would know it has flagella. Did it tell us the flagellar arrangement? Did we see the flagella? No. So you can't see the flagella, but it's implied that at least one is there on your cells if they are motile, okay? So we can see motility, but we can't see the flagella. We could see the cell shapes, right? And kind of a little bit of the arrangements when we looked at those. And we also could potentially see Brownian motion, which is when the liquid is moving the cells around. So those are things you can see on the wet mount. I feel like that pretty much covers everything you're going to need to know for the wet mount. There's not a lot to know for it. All right, the endospore stain, again, malachite green is your primary stain. The steam and the heat is your mordant. What was the purpose of the paper towel? Just to keep the stain on the slide, so it was less likely to drip off. A lot of us still had it drip off, and that's fine, we survived it, but that was really just the point of the paper towel, was to help keep the stain on top of the slide. And it didn't serve any purpose in the actual staining procedure. We could have done the staining procedure without the paper towel. But the steam was our mordant, our decolorizer was deionized water, and our counter stain was safranin for that one. All right, any questions about the stains? Okay. There's a lot of questions on the exam about the stains. We'll also be talking about like microscope parts and their functions, stuff like that. But now we're going to talk about ELISA, which is the one that we just did recently, so it should be fresh in our minds. What does the E stand for? Or how I told you, you're going to have to know what ELISA stands for. I would know what the whole word stands for. And again, you won't have to write it out. You'll have multiple choice. What does ELISA or ELISA? I don't care how you say it. So it is enzyme-linked immunosorbent assay. That's what the ELISA stands for. Enzyme-linked immunosorbent assay. Okay. So it stands for enzyme. The E stands for enzyme for that one. All right, what does an indirect ELISA test for? What is it testing for? So at least we're between the two major choices. The two types of ELISAs that we have to know about are the direct ELISA and the indirect ELISA. And the one we did in lab simulated an indirect ELISA. And so that was looking for antibody. Does the patient have the antibody? The direct ELISA tests for the antigen. Remember, the antigen is a marker of the pathogen. So it's basically like asking, does the person have the bad guy directly? Whereas the indirect, the question is, has the patient ever seen the bad guy? Is there evidence that they've seen the bad guy? So we're not looking for the bad guy, but whether they have seen him. And so that's an indirect way of approaching it. So the question is, does the patient have the antibody? And really quickly, if you're checking for antibody, what do you need to have as you're known? You need to supply antigen to see if the antibody is there. So we have to start with that. And again, if we were doing a direct ELISA looking for antigen, we would have to supply antibody. So have that reference as well. Okay. All right. For both of them, color change is positive. So it's the same in the end, but what does that really tell you about your patient? All right, so principles of how the assay works. It depends on antibody antigen specificity. Okay, so that antibody sees that one antigen. You're not actually gonna, it's very specific and it's very sensitive for those terms, right? What are antibodies and what are antigens? And antigen is something that your immune system could see as non-self, identified as foreign. An antibody is a protein that your body makes in response to the antigen, the thing that was foreign, okay? We kind of already covered indirect and direct. How does the color change happen? If your patient is positive, then the secondary antibody that's linked to the enzyme can bind, period. Whether you have, it doesn't matter what type of ELISA you're doing. That part is the same. So if your patient is positive, the secondary antibody with the enzyme will bind so that when you add the substrate, the enzyme will cleave it and cause a color change, okay? Keep in mind that whenever you do an ELISA, you also are relying on your positive control and your negative control. So if your positive control and your negative control didn't work out, you can't use your results. So if I ask you, what does a properly performed positive result look like? You can't just tell me, oh, well, the whales would change color. You have to also tell me what your positive control and your negative control were doing, because we can't rely on the data unless that stuff is there. So keep that in mind as well. Rapid testing, I'm not going to test you about it on the exam, okay? But it works on a very similar principle, right? And so when you do home testing with COVID, it works on a similar principle. It's just not in wells, and it's not as quantitative. All right, moving on to the DNA stuff. This is that transformation simulation and that DNA knowledge sheet that we had. Typically ask kind of basic questions about it. So what does PCR do? We had a question like this in our quiz for unit 2. I think it was chapter 8 quiz. And if you're ever taking an exam, you're like, what do you mean by amplify? And I always just tell you that it means making copies. So that's what that means. So it literally, PCR stands for polymerase chain reaction. And the whole purpose of it is to just make copies of DNA. Remember the 3 steps of PCR: denature, anneal or priming, and then elongate or extension, however you want to see it. But you denature to separate the strands, you bind your primers. If the primer is bound, then you will extend A's to T's and C's to G's with your DNA polymerase. So that's the way that PCR works. And if I were to ask you, if we start with 1 DNA, how many do we have after 1 round? 1, 2, and then that would be 1 round, right? So after a second round, how many would we have? 4, and that's the second round. And how many after 3? Okay, so that we're clear. Obviously, I've said it before, but I'm gonna say it one more time. That type of question is gonna be on the exam. It will always start with 1, but it will go, I will choose anywhere, and you've been warned, between 1 and 6 rounds. And how many would you have after that number of rounds? Okay. So I say, how many would you have after 1 round? You would say 2. You have enough context for that? Okay. PCR. Which DNA fragment is largest? Your choices here. I spent a lot of time giving a little preface for this one. But again, when it's typed out, it looks a little bit different, and we sometimes aren't paying attention to everything that's on the screen. Which one is the largest? All right, so most of us got this one. 35 kilobase pairs. So we have 1,200 base pairs, 4 kilobase pairs, which is 4,000, 35 kilobase pairs, which is 35,000, and 100 base pairs, which is just 100 base pairs. So here, the largest fragment is 35,000, 35 kilobase pairs. All right, which of the following DNA fragments would travel the farthest on an agarose gel? It's the same choices. Which one will go the farthest, though? You could think of it as traveling the farthest or traveling the fastest, but they mean the same thing. All right, so 100 base pairs because it's the smallest one. The smallest ones travel the fastest and the farthest. The largest ones travel the slowest and make the least progress. They get trapped in the gel. Okay. And remember, which direction would DNA migrate in the gel? Towards the positive. So there it's negatively charged, so it'll move to the positive. Okay. Just another reminder about that. All right, what type of cells are capable of taking up exogenous DNA? There's a little bit of context for this one. All right, so most of us are getting this one as well. So this is if you're able to take up exogenous DNA, cells that can do that are called competent. And the process through which they do it is called transformation. So this is that transformation experiment. I didn't tell you outright that it was transformation, but we had to kind of infer, okay, this is that transformation process. If you can go through transformation, then you are a competent cell. You don't have to be E. coli. You don't have to be a pathogen. Not all cells can do it. Only competent cells can. All right. Good. We're doing good so far. All right, DNA stuff. We talk about DNA fragment sizes based on base pairs. Be sure you're paying attention to the Ks or Ms if there are any present on there. That kilo means 1,000. Agarose gel electrophoresis. I feel like we kind of covered that pretty okay. And how PCR works. Bacterial transformation. Very straightforward with that. It's mixing competent cells with plasmid, and plasmid is DNA, right? Just extra genes of DNA in a circle. So we mix plasmid with cells. We heat them to open them up so that they'll take it up, and then we'll cool it down to seal it back in. And that's the transformation process. Be aware of what the results would look like on each of those media types if you have a plasmid and if you don't, and which genes are you expressing on the certain media types. So be aware of that as well, because those are questions that I do ask on the lab midterm. So you've been warned about that. And plasmids, remember, they are DNA. All right. Any questions about transformation of the DNA stuff? I didn't pause for Eliza. Any questions about that either? Eukaryotes. We have our drawings, quote unquote, but you guys got to take pictures of what I showed you on the overhead. So let's see if we can answer some questions. Already, if we look at just this picture here, even though it's not the same color as what we saw in ours, we should be able to identify that picture there as looking a lot like our penicillium. Remember that one. All right. Again, yours will be the same slides as what we saw in class, so don't stress too much. Which organism is pictured here? It's not as vivid as the one we had in class, but this is the same stain. It is from one of the slides in class. It's just less vivid of a color. This was penicillium. Good. Okay, so that one's pretty easy. What would aspergillus look like? Green, right? And like dandelions. Okay. Definitely we identified those, know the difference between those two. All right. What is the name of this organism that's pictured here? I say purple snakes around some round thing. That's there. Okay, but we should be able to identify that one. That is trypanosoma. Trypanosoma. So you do not need to, if we didn't recognize it in the class, you don't need to be able to recognize it in the class, right? So be aware of which ones we went over and be able to identify them by site, because it will be questions like that. Like I'm just going to put a slide up and be like, what organism is it? Or if I was asking about that, I could also ask something about like, what was this this is a protozoa, is it a trophozoite or a cyst? We didn't look at any cysts, did we? So automatically if it's going to be protozoa, it must be a trophozoite. So even without having any reference on that, for that example, you do need to know the difference between a trophozoite and a cyst practically, like what the definitions are. You'll need to know that for the lab exam. Trophozoite, active, multiplying, reliving. Cyst, dormant and inert. It's the way that it gets into the body, right? But it's what multiplies is the trophozoite. So this is the trophozoite form of Trypanosoma gambiense, right? Remember that Enterobius vermicularis was one of the helminths that we have to know, but it was the eggs that we had to know for. It was the oval-shaped eggs. We didn't have to look at Rhizopus, and syphilis is a bacterium, and we haven't looked at that. Okay. Well, the eukaryotes, know the organisms that we took the pictures of. It wasn't that many, so know those ones. Basic major differences between them. Important structural and reproductive differences. Again, we're not going into a lot of detail on that. Just know them by sight and the general, like, oh, this, if you look at it and you're like, this is a helminth egg, or look at it and say, this is a protozoa trophozoite, or look at it and say, this is a fungi. I'm expecting that. And what did I expect you guys to be able to identify for the fungi? That they are what we saw with our penicillin, for example. That was an image of the conidiophores, right? So know that they are conidiophores. That's an asexual reproductive mechanism of fungi. Okay. And you do need to know the transmissions of the organisms as well. One of them I will test you on. I'm not going to tell you which one. But you do need to know the transmission methods of those guys. All right. Any questions about the eukaryotes before we move on? Bacterial growth stuff. Media concepts and culture techniques. Cell shapes and arrangements and stuff. Okay, which of the following is not a term used to describe colony morphology? This one's hard for a lot of people. I said that you have to at least be able to recognize what terms were colony morphology terms and which ones weren't. This one is difficult for some people. But we'll say it's just one. Yes, so hyphae is the one that is not a colony morphology term. So all the other ones could be. Plateau is one that you could see. It's basically looking at the elevation. Irregulars, looking at the shapes, the edges of the cell, the margins. Convex, again, it's going to be an elevation, it's just rounded. Punctiform was tiny little dots. That was the overall shape of the guys. And rhizoid is also basically like a surface texture type of a thing. Is it branching a lot off? At the microscopic level, if individual cells of our fungi are branching off and creating each other at the microscopic level, those are called hyphae. But that's microscopic, not macro. Okay, so remember that colony morphology is macro. And anyways, this was a harder version of definitely the one that I have on the test. It's going to be easier than this one. Okay, so this one is a little bit harder. Not all of the questions on this review are. I feel like most of them are pretty similar to what you're going to see at difficulty-wise compared to the exam. But this one was a little bit harder. But typically what I will do is you'll have a list of about 5 terms, and I'll say which one is the colony morphology term. The next question will be which one is the cell shape term. And the next one will be which one is the cell arrangement term. You get what I'm saying? So it's going to be the same list on 3 different questions, and you're going to have to identify out of 5 terms which one applies to what situation. Okay. If you study up on the other stuff, that's not meant to be, that's meant to be an easy question when we get to that on the test. All right, what is the name of this type of culture? It says on it that it is agar. Okay, so this is a solid culture. We had these in our black top tubes that we used occasionally. On an exam, I would give you like one that you could pick up and see if you needed to. you would see that it is solid, and then it has a slant. It's quite literally just a slant. Don't read, again, this is not a trick question. It's meant to be easy, that type of a question. So we have 3 types of solid media and 1 type of liquid media that we have to be aware of. The solids were the auger plate, the slant, and the butt. We haven't seen a butt yet, but we've talked about it, right? And then the broth is the liquid one, the only liquid one. It's straightforward. Don't think too hard about that. What do we add to media to make it solid? Auger, right? We agree on that. And where does auger come from? Algae. It's derived from algae. Okay. It's a solidifying agent. All right, how is this organism isolated? What technique did we use to isolate this organism? We did them in class a long time ago now. All right, so this is the streak plate, pretty easy. Ours was the quad streak plate. And in which quadrant do you find the most single and isolated colonies? Which quadrant for the quad streak? 4. Okay, so we're good on that. All right. So I give you a picture of a quad streak plate. We could say, okay, so it's meant to be easy. Like I said, some of the questions are easy. All right. Don't need to study quad streak plates. Don't waste your time on it. You know that one. Which arrangement do you observe here? Based on our choices, what arrangements do you observe? Don't read too much into it either. On the exam, it's not multi-select. It's just one answer that'll be right, okay? And I'll tell you, yeah, so I would have gone with this one. I would have gone strepto. Interesting. Just saying. And this is why we see chains. Would we agree we see chains? So this is maybe where we need to study a little bit. Y'all still having problems with staphylo and strepto. You got to get it down before you get to the exam. Bunches. If you see that it is generally overall lots of bunches, then that would be staphylo. If it's generally overall chains, that would be strepto. And on the exam, if I were to give you this, I would say, what is the general overall arrangement? Could you argue that maybe here it looks like clustering? You could maybe argue that, but you could also, I would say, like, come on, let's use common sense here. That's chains that are folded back on each other. That's not a cluster. Okay? So when I ask for general overall, I would say that this is chains. Chains is strepto. Bunching is staphylo. Okay. And this was not asking for the cell shape. I didn't need bacillus. I didn't need cocci here. Those are cell shape questions. You understand the difference? Okay. Let's try the next one. What is the name of the shape of these bacteria? So now we're looking at the cell shape. Quietness of this. So these are bacilli. These were rod-shaped bacteria. Remember, cocci are going to be little circles. If you're at all feeling like they feel almost circle-like, but they're kind of squatty, then those would still be bacilli. Okay. So anyways, like I said, you can ask me too. Like if you get into the exam and you're like, I think these are round cells, I can say, yes, I think they're round also. If you're ever confused about that. And I'm not going to give you on the exam, I'll give you something. To me, this is very obviously bacilli. Expect that obvious on the exam. Okay, so if you look at it and you're like, I think these are kind of round, lean into that. It's not meant to be tricky on the exam. Okay. Moving forward, when we look at our unknowns, that stuff might get a little tricky. Okay. All right. So colony morphology, be aware of what terms are colony morphology terms. So on the previous question that we had here, one of our choices was umbonate. Umbonate is a colony morphology term. That's all you need to know about it, is that it is a colony morphology term. Okay. So be aware of that. The different media types, which we already went over. Again, very basic concept of the media types when I say that, okay? Auger plate, slant, butt, and broth. Know what those are. We are not going to test you over differential and selective media. That's in the next part of the class, okay? Isolation techniques, we only have to know about the quad streak plate, so be aware of how that works. Cell shapes, you only need to know bacillus and coccus. And then cell arrangements, there's only 3 you have to know. Staphylo, strepto, and no arrangement. That's it. Those are the only ones that could possibly be on the test because those are the only types of bacteria that I have. Okay. Any questions about that? All right. Microscopes and how to use them properly. First question, if you observe a specimen using a 40x and you have a 10x ocular lens, what is the total magnification? This is one that sometimes we'll get people because we're not checking on how many zeros or whatever. So if you're looking at total magnification using a microscope, what usually will happen is like on one of your other slides that you'll be looking at in a microscope or like a stain or a eukaryote or something, I'll just tack on a question that'll be like, what is the total magnification as it is set up and you're looking at it? And so you can look at your objective lens, and all of our ocular lenses are always 10x, but they say it on there too. So you would know to multiply whatever your objective lens is by 10, which is how you got 400x. So just a reminder of that. The ocular lens and the objective lens. You do need to remember that the objective lens generates the real image and the ocular lens generates the virtual image. I wanted to touch on that before I forgot. All right, which objective lens do we use for oil immersion? You can do this both ways. If you're doing oil immersion, it must be this lens. If you're using this lens, you must use oil immersion. And it's the only lens that we use with our oil immersion on our microscopes. So 100x, 100x. You shouldn't be using oil on the 40. In fact, the oil on the 40 can actually damage it. All right, so be able to calculate magnification, understand generally how the microscope works. Again, so I might put a piece of tape on a microscope part, ask you what is the part, and then what is its function. Okay, so that type of question is common on the lab midterm. So be aware of all the parts and all their functions that we've talked about in the class, how to use all of the parts. I'm not going to make you show me how to use it, but again, you should have reference for that in case I ask about it, okay? Understand why we use oil immersion. Remember that oil immersion works to reduce light scatter, also known as reducing refraction. You'll have multiple choice on this test. We've had that question already on the Unit 1 exam. So I feel like we did pretty good on that generally for that one. So we'll probably have that down pretty well. Any questions about the microscopes? Kirby-Bauer. All right, so the assay that we did for antibiotic susceptibility. This is called the Kirby-Bauer assay. What is the name of the media that we used for it? Mueller-Hinton is the media. So the actual plate is Mueller-Hinton. The assay that we performed is called the Kirby-Bauer assay. I say it a lot because guess what? It's gonna be a question on the test, okay? All right, so based on this Kirby-Bauer assay that's gonna show which antibiotic is the bacteria least susceptible. So least susceptible, same thing as least sensitive. Don't overthink it. All right, so it was E. We knew it was E because there was no zone of inhibition about it, right? So again, don't think too hard on it because what will we document that zone of inhibition as if we were going to be measuring these? E's would be 0. Just mark it down as 0, you don't even need to measure it. And so we're looking for the one with the smallest zone of inhibition, and the smallest one is E, 0. Okay, makes sense? Any questions about that? Know what a zone of inhibition is and have an idea of how it relates to the minimum inhibitory concentration, okay? That you can measure the zone of inhibition and that you could calculate the minimum amount needed to inhibit to use the dose, okay? Mueller-Hinton auger is the auger that we use for that first step of the Kirby-Bauer assay. Remember, there was a second step where we took a sample from that Mueller-Hinton around the zone of inhibition and then plated on the next plate. If it grew on the next plate, that meant that that antibiotic was bacteriostatic. If it didn't grow on the next plate, then that meant it was cidal. Do we agree? Any questions about that? Okay. Because I think this is pretty much it. All right, and that is it for the lab midterm review. It is 10 o'clock, and I'm not going to keep you any later than this. So that is it for the lab midterm review. We will almost certainly have the lab midterm on Wednesday. Again, if something weird happens with my other classes, I'll post an announcement. So look out for the announcements for that. And wait till you hear from me. Either way, even if we don't have lab midterm on Wednesday, we will still have lab on Wednesday. So I'm kind of in the air on whether or not we're having the midterm, but be here on Wednesday at 11. Good? Okay. Good job, guys. For what it's worth, if you guys want to know what the average would have been if this was the exam. If this was the exam. Anytime it wants to load. Okay, it didn't want to, I guess. Yeah, it's not loading. Okay, never mind. Again, I don't know what's going on with this. It's just not loading it. Okay, cool. Keep hoping and keep crossing my fingers that it's gonna load. Yes. So if you use the link and you submitted it, it takes at least 45 minutes to an hour for them to validate it. They should get to it today. So just look out for that. And
they had access to
the app. Yeah.
And the online.
Okay. Yeah, that's probably just because your cookies haven't reset. But it's like once if you were to try to open lockdown browser, it probably wouldn't work. Like you have to like relog in, and then it'd be like, oh, your password doesn't work, and then yeah.
So do I have to reset it then?
You will. I mean, that should start that for you. And so my understanding is that just doing the validations of the identities, and then if they need you to reset your password, you'll get another prompt for that.
Okay.
But yeah,
right
now they're just doing that. Yeah, and we'll see how it goes. Oh, I just didn't load my. Makes me sad.
I did my verification on Thursday, but I haven't had to like reset any passwords, and I'm still getting like emails and everything's. Is that just because I'm an employee?
It could be.
Okay.
Yeah, it could be that. It could be because you're an employee. It could also be a personality. So
Okay.
if this was the exam for the lab midterm today, I don't know why it's so slow.
It's tired so much.
Yeah, I understand that. Anyways, yeah, maybe it's because you're an employee of 67. So not great. I expect by the time that we have the exam, that'll be up to at least 75. People usually do a little bit better on the lab exams.
Is that exam 50 questions or 100 questions?
It will be 50.
Okay.
It's just 50. Yes. All multiple choice, 50 questions. I'm trying to think if there's anything else. No, otherwise it should be, yeah, and it'll be on Canvas, like in lockdown browser, like just like we do our like lecture exams. We could be walking around with them from station to station.
Okay.
Yes.
Okay. Thank you.
Yes.